Today we will be exploring the impacts of gravity or the lack thereof gravity at the cellular level. We will be doing this by exploring the journal article published by the team of scientists, academics located at universities around Australia and China. This article is Modelling the Impact of Microgravity at the Cellular Level, Implications for Human Disease. In this video, we will be analysing the findings identified in this piece via several studies, as well as the impacts these such findings may have in the wider community, along with how the findings and/or observations made in this article relate to both the big ideas of science and about science. Please find the article cited in APA 6 formatting, along with the link to the article in the description. In the photo here, we can see the two NASA astronauts, Suni Williams and Butch Wilmore, who have recently returned to Earth after being unexpectedly stranded on the International Space Station for 9 whole months, well beyond what their original mission timeline was set out for. The original plan was for a shorter stay. Their extended mission resulted from technical issues with their spacecraft, which forced NASA to delay their trip back to Earth. Living in microgravity for such a prolonged period has significant effects on the human body. Without gravity, muscles and bones weaken because they are not bearing weight as they do on Earth. The astronauts have experienced muscle atrophy, bone density loss, and fluid redistribution, which causes facial puffiness and pressure on the eyes. Their cardiovascular systems have also adapted differently. NASA will closely monitor their health, providing daily exercise routines and medical evaluations to manage the effects of long-term spaceflight. Their situation provides critical data for future deep space missions, such as planned journeys to Mars. Although these effects or implications of microgravity are currently reasonably well known, the effects at the smaller cellular level are not so well understood. However, these effects are just as important as those previously stated, as they will help us to better understand why such effects are observed in those exposed to microgravity for long periods of time. Mechanobiology studies how physical forces like gravity influence cell behavior through mechanotransduction, converting mechanical inputs into biochemical signals. On Earth, gravity helps maintain cell and tissue health by applying mechanical stress. In microgravity, this stress is removed, disrupting normal cell function. Simulating microgravity allows researchers to study these changes, which can accelerate conditions like osteoporosis and kidney stone formation. Although the effects of microgravity are well documented, the exact cellular and molecular mechanisms remain unclear. Understanding how cells adapt to weightlessness is essential, as it reveals insight into human physiology and guides further research into space-based mechanobiology. This knowledge could lead to improved treatments and technologies for both space travelers and patients on Earth. Simulating microgravity on Earth is challenging and expensive. True weightlessness, like that in space, is hard to replicate due to Earth's constant gravity. Ground-based methods such as clinostats, random positioning machines, and parabolic flights can mimic some effects but have limitations in duration, scale, or accuracy. Parabolic flights provide only brief periods of weightlessness and are very costly, while space-based experiments offer the most accurate data. They require significant funding, strict safety measures, and lengthy preparation. These challenges make it difficult to study microgravity's full effects on biology. Researchers must carefully balance scientific goals with practical limitations, making microgravity simulation a major hurdle in advancing space sciences and medical research. Microgravity significantly affects the cytoskeleton, which helps cells maintain shape, move, and respond to mechanical signals. In weightlessness, cells often change in shape, size, and adhesion due to disruptions in microtubules and actin filaments. Microtubules lose proper organization without gravity, while actin responds vary. Some studies show reduced actin, others show increased stress fibers. Microgravity also weakens focal adhesions, reducing cell attachment and altering key signaling pathways. These changes impair cell movement, survival, and communication. In bone cells, this leads to faster bone loss, similar to osteoporosis. Cartilage cells also suffer, producing less extracellular matrix and showing early signs of joint damage. These effects highlight how altered mechanical forces in space can impact tissue health and disease development. Microgravity accelerates bone loss by disrupting the function of mechanosensitive bone cells like osteocytes and osteoblasts. These cells rely on the cytoskeleton and microtubules for structure and signaling. In space, cytoskeletal collapse and reduce focal adhesions, impairing key pathways, especially the bone morphogenic protein axis. This disruption increases sclerostin, a protein that inhibits bone formation, leading to bone resorption and density loss, similar to osteoporosis, as previously mentioned. Insights from spaceflight and microgravity studies contribute to the development of Evenity, an FDA-approved drug that blocks sclerostin and reduces bone growth. Cartilage cells also show reduced matrix production and signs of degradation in microgravity, but more research is needed. One study exposed mice to 30 days of spaceflight to examine these effects on joint cartilage. Microgravity significantly affects the immune system, especially T-cell activation, which is crucial for fighting infections and cancer. In space, T-cells show reduced activation and altered signaling due to changes in the cytoskeleton and gene expression, weakening the immune response and increasing disease risk during long missions. Microgravity also impacts cancer cells in diverse ways. Some become more aggressive and treatment resistant, while others become less viable or more sensitive to therapy. These varied effects offer unique insight into cancer behavior and immune function. Studying how T cells and cancer cells respond in space can lead to improved therapies for immune dysfunction and cancer, benefiting both astronauts and patients on Earth in the future. Microgravity or near weightlessness in space significantly affects cells, altering growth, gene expression, and metabolism. Without gravitational stress, bone and muscle cells weaken, and fluid behaviors within cells change. These effects emphasize the importance of cellular organization in organisms and how they adapt to different environments, which is related to the big idea of science, where organisms are organized on a cellular basis. Studying microgravity helps scientists design better space habitats and medical technologies to support astronaut health. It also benefits Earth-based medicine by advancing treatments for conditions like osteoporosis and muscle atrophy, as previously mentioned. This research connects biological understanding with engineering solutions, showing how science and technology work together to solve challenges in both space and healthcare, which is related to the big idea about science of the knowledge produced by science is used in engineering technologies to create products to serve human ends.